Hi, my name is Philip Furco. I'm a senior scientist here at EAG Laboratories, and I work a lot on chemical characterization studies. And today I'm going to be talking about interpretation of recent developments in FDA guidance for chemical characterization. So it's going to be a summary of some notable developments in FDA expectations for chemical characterization studies. And it's going to be talking a bit about a recent draft guidance document and other activities from the FDA. So to begin with, I'm going to begin with the background on these types of studies and some of the work that's been going on and expectations from the FDA. And then I'm going to dive into three deliverables. The first one's the draft guidance review, which came out last year. Next one will be the chemical list for analytical performance. That's an ongoing project within the FDA. And then also discuss the ASCA workshop that was held late last year. I'll wrap up with some conclusions. So there's three deliverables that we're going to be talking about today. The first one is the chemicals list for analytical performance, which came out in July of 2024, at least the beginning of it did. I'm going to talk about the draft guidance, chemical analysis for biocompatibility assessment of medical devices, which came out in September of 2024, as well as the ASCA workshop, which was in November 2024. So these three things are related to each other in that they all touch on chemical characterization, but I'm not going to discuss them in the chronological order that they arrived because it's not really the best way to tell the story. Instead, I'm going to start with the draft guidance and then discuss the subsequent ones, that is the chemical list for analytical performance and the ASCA workshop later, because I think it explains things a bit more clearly to go in this order. So ASCA is actually an accreditation scheme for conformity assessment. There is such a thing already in practice for in vitro testing, but the FDA is discussing the best way to implement that for chemical characterization. So next, let's talk a little bit about the background. So ISO 10993 Part 18 describes chemical characterization for medical devices. So this is within a risk management process, and it's described by this ISO standard. Historically speaking, EU MDR has accepted Part 18 studies. If you follow the standard and you're applying it as it's written, and you submit to the EU MDR notified bodies, you're not going to have any issues as long as your device is deemed to be safe. Many of you may know that there were deadlines that were put in place, and then these were postponed. But this was based merely on the volume of work. It wasn't about technical challenges or issues with the standard. So, you know, what I've described is pretty much that you could do this work, you submitted it, the EU would accept it. There weren't really too many issues. This is a little bit in contrast with the FDA's approach to ISO 10993 Part 18. So the FDA partially recognizes ISO 10993 Part 18. There are a few sections that are taken exception to or are not accepted. So these center around things like the analytical evaluation threshold, the grouping of compounds, and also the calculation of uncertainty values. The document in which this revision, or excuse me, in which the exceptions are described in terms of the FDA accepting Part 18, it describes what they are, what those sections are specifically that they take exception to. It also provides some literature references, but it doesn't really say what the solution is or what the FDA expects in place of that. So those details have largely been communicated through reviewer feedback. Also, the alternative approaches that would be acceptable, and just generally the interpretation, that's all come from reviewer feedback. And that's a one-on-one kind of interaction, right? This is an individual project that a study sponsor may be receiving feedback on. And so they'll say, in this particular project, we would like to see this instead. And it's good because they are describing the path forward, but the difficulty there is that it's sort of one-on-one, and not everyone is able to see all of that information. It's really only people that are involved in that particular project. So the draft guidance and the accreditation strategy discussion, all of this is clarifying expectations, and this is really a positive development. So we no longer have to depend on having recent experience with submission and potentially pre-submission or discussions back and forth with the FDA to understand what exactly is expected. Those are going to be committed into writing, and ultimately that's a positive development. So with that in mind, let's now move on to actually discussing the draft guidance. So one of the first sections I want to talk about is the scope section. So it has the typical statement that these are recommendations, that departures are allowed with justification. It is in fact for non-targeted and targeted work for biocompatibility endpoints. You know, we often think of Part 18 studies as non-targeted, but there is the potential for follow-up work or you know, additional work that is targeted. There are devices that are excluded from this guidance. So these are ophthalmic devices, respiratory devices, and dental materials. The reason that these are excluded is because there's actually already vertical standards within ISO that address the same concerns about biocompatibility, but take into account particular details of these types of devices. So it's not that these things aren't being addressed anywhere, it's just that they're being addressed in a different standard, and this draft guidance is not necessarily going to address that. It's questionable, maybe down the road, there may be some bleed over that the expectations from Part 18 may influence FDA interpretation of these other standards, but as it currently stands, that's not baked into this document. Last thing in the scope that's worth mentioning is, as always, if you have a challenging device, you should consider the Q submission process where you can get FDA review and buy-in or input on your plan prior to execution. The next one to move on to is the section discussing the devices for testing themselves. The FDA is, again, restating the expectation that is in the original ISO standard, but is really important is that we're looking at the final finished form, including sterilization and packaging, when we're testing devices for chemical characterization. This is really important because the schematic below shows the whole process of a medical device being prepared to the point where it's used on a patient, right? There is the initial manufacturing. There's also going to be cleaning steps, packaging steps, as well as the sterilization. And each of these steps has the potential to add or remove extractables and leachables and change the actual leachable profile that a patient's exposed to. So it's really important that the devices that we're testing actually go through that process. Most people know this, but it does sometimes come up. So it's important to be aware of this. Why it's important, in addition to the patient safety aspect, is that this can often impact the timeline of a study for a study sponsor. You can consider, you know, if you're in a phase in the product development where manufacturing is not being done on the line, there can be some lead times there. Sterilization can also be an issue. If you have a third party that's performing the sterilization, there's some shipping and possibly backlog at that sterilization facility that can impact your timeline as well. And that's important because chemical characterization can often be on the critical path for biocompatibility assessment. They also specify that if you're going to use a device class argument, that is that you're going to have identical devices of multiple sizes that are used in the same fashion. You don't necessarily have to test every single one. If you can make an argument that they're all being manufactured and handled the same, meaning cleaning, packaging, sterilization, then you can test the largest device and use that to justify the exposure that's expected from the other devices. It's important though, even though that this is acceptable, they do state that extrapolation of surface area is not considered the worst case scenario. And that is important because historically we have seen that where maybe devices aren't available for that largest device type in a class, and so extrapolation arguments are made. That is, it's kind of clear that they're not really going to be accepting that for much longer, if at all, at this point. Next, they talk about one of everyone's favorite topics, swelling within devices. So they do mention destructive versus non-destructive swelling. They don't really define them. The definitions that you see here are actually just ones that I've taken kind of from literature and general chemistry expectations, right? And that's it. And a non-destructive swelling is going to be a reversible process. And what it's really going to do is it's going to increase extractables. As you swell a material, it allows for more exchange of solvent in and out of it, and we can extract more out of the device. So we can go into a more exaggerated regime of extraction, but we're not necessarily compromising the device. Destructive is going to be irreversible. So it may swell to the point where it ruptures. You can have degradation products forming, and the device integrity is compromised. So what I believe is going on here is what they're really saying is that they don't want to see people using swelling as a justification for avoiding doing a certain solvent class. And we'll take just a hypothetical device, and I can kind of walk you through why I think this is this way. So if we have some sort of permanent implant device, it could be polymeric. For this hypothetical case, we'll say it is. And we do solvent compatibility studies, and we show that all non-polars cause this device to swell to the point where we say that it's destructive, or maybe some are non-destructive, some are destructive. But we do this and we say all of these show unacceptable swelling, right? And we're not going to define that too tightly. But what we may do is we may make the argument, well, we did the test, we did a solvent feasibility study, and we couldn't find a suitable non-polar. And so we're going to omit that from our subsequent analysis. I think the FDA's concern here is now that we have a device that we're only going to subject to 2/3 of the testing that we would have otherwise, and that the standard expects for a permanent implant, right? So we would proceed with the polar and mid-polar because those worked. We didn't have the swelling issue, but we've just dropped off 1/3 of the data that we're actually going to review and use to support the biocompatibility assessment. And I think that's why they're really trying to close up this loophole where we test in a few solvents and we say we can't find one, and so then we stop. And this is kind of reflected in this section because they do say that. They say if you really can't find a solvent that's acceptable and it's one of the solvent types, meaning mid or non-polar, that's needed for this type of device, then chemical characterization may not be appropriate for demonstrating that biological endpoint and biocompatibility. So this is kind of closing up, and I think it's important because if people are operating on legacy ideas, you may end up in a situation where once you've submitted, the FDA has issues with a decision to omit a solvent. There's also discussion around particulates, and they are clarifying their expectations on what they want to see. I don't think it's a surprise to anyone what they want to see, but it's a little bit clearer so people know exactly what the goal is. And they also clarify some things on NVR determination, and these are pretty important as well. So what they're really looking for is characterization to determine the likely source and chemical composition. This does start to fall in line with TIR 42, which is a technical report from Amy that kind of begins to discuss particulates, and these are things that they're looking at as well, chemical composition, because that is going to impact the risk assessment. They also draw a clear difference, line between particulates and precipitates, right? So particulates are true particulates. These are solids that are being broken off or rinsed off the device during the extraction process. Precipitates, because we're using elevated temperatures and aggressive solvents, we could extract something that is somewhat soluble in the extract solvent, but only at elevated temperature. And then as those cool down after the extraction, right, so we go from 50 C extraction to room temperature, we could have those things that are dissolved in warm solvent then begin to precipitate out. And the reason they're drawing this distinction is that something that breaks off the device under the extraction process is something a patient could be exposed to, right? Whereas a precipitate, presumably it's going to be removed by, it's not going to build up in the tissue to the point where it precipitates out necessarily. So they're looking for that. What's an artifact of the extraction process and what is truly particles coming off of the device? Because they're trying to answer the question, could tissue be exposed to particulates from the final finished device? Right, and that's the concern around particulates, and it's those effects as particulates they could have. They encourage you to analyze particulates per ISO TS 10993 Part 19, which goes into discussion of the various approaches to kind of answer analytical chemistry questions around particle identification, size, and these sorts of things. The last thing they do is they discuss the impact on NVR determination, and they make it clear that particulates rinsing off a device are not considered part of the non-volatile residue. So what they're suggesting instead that needs to be done is that solution containing the particulates needs to be filtered to remove those, and the NVR be conducted on the truly soluble portion because those are the actual extractables. Naturally, if you're doing a sample handling like that, the FDA is also going to want to see some demonstration that that approach is suitable and that you're not removing relevant extractables from the analysis. Also discussion on exhaustion in the draft guidance review. So again, we're talking about the process of taking our extractions, taking some of them, drying them all the way to dryness to remove volatile components, right? So the solvent as well as things that are volatile that are extracted. but we're looking at that, we're looking at those non-volatiles to demonstrate that we've exhausted the device in the extraction process. So there's been a lot of different approaches around this. I would say there's been some creativity, historically speaking, especially in instances where there are a limited number of devices. And they're starting to draw some lines around this and say that they really need to be conducted in a certain way to make sure that we are getting good results. The FDA really, they prefer the gravimetric demonstration of exhaustion, and it is good because it is kind of an orthogonal method, right? If we use different chromatographic techniques, right, LC, GC, there's a potential that that particular technique will not see something, whereas the gravimetric approach is really a universal detector. If it has mass, we will see it that way. So it's a nice orthogonal cross-check. So what are these recommendations? They want to see a full volume of extract used in that non-volatile residue determination. So what they don't want to see is taking an aliquot from another extraction and using that. And they also want to see them performed in triplicate, right? So again, it's the whole capturing of variability that triplicate analysis allows us to do. They also want to see exhaustion demonstrated for each solvent. This is reasonable, right? We do know that solvent's going to behave differently, and the extraction profile is going to change based on the solvent type. And so it really doesn't make sense to pick one solvent and use that one to justify the extraction or the number of cycles to reach exhaustion, right? We can easily imagine a device where water would extract very little or nothing, whereas the mid or non-polar could extract a lot more over time. And so if you depended on water, you would miss a lot of what was coming out of that device. The last statement is they want to see a justification if exhaustion cannot be demonstrated by a gravimetric approach. The example they give, which I found to be interesting, was polymer devices in water. I think another class of device that everyone can readily intuit that would do this is metallic total implants, right? These devices are very clean, they're impermeable, they don't have additives in them. So what you would probably see is minimal surfactants or processing aids coming off them, if anything at all. And so it'd be difficult to demonstrate exhaustion. It remains to be seen what that will look like. I do know from discussions that we've had with the FDA that if exhaustion cannot be demonstrated gravimetrically, they are interested in seeing it demonstrated chromatographically, meaning going through the results from chromatography and showing that you're getting less than 10% in your final extraction versus the first one. This makes sense because it's more sensitive, but you do lose that orthogonality, right? Because you're demonstrating exhaustion by the same method by which you're actually doing your characterization. So it's not as universal. There's also a very oblique mention of data deconvolution in the draft guidance, but if you're in touch with the industry, you know that this is really where things are going. So data deconvolution is, just very briefly, a way to take a pretty complicated extractable profile that's been analyzed chromatographically and separate it out into its constituent components. So on the left, we just have an example chromatogram. So it's been kind of split out. Along one axis, we just have the time of the analysis. In the z-axis, of course, we have some sort of abundance, and then we have the m over z along the y-axis, so going back into the slide. And so what you can see is that a single injection is pretty complicated. There's a ton of compounds coming out here, different times, they're overlapping, that sort of thing. And what we can do is we can apply this data deconvolution approach. So basically creating a bunch of extracted ion chromatograms to fish out lower abundance, but still very important signals, because these can be above the analytical evaluation threshold. So on the right, we have an example of this, right? The black trace over everything is just the base peak mode. And then we have some blue peaks, which are extracted ions that we're actually pulling out of that total ion chromatogram. And so we can see the large ones, and if we were just using a base peak mode, that's what we would see. But you can also see that there's some smaller red peaks sort of buried here. We might see a shoulder, we might see a little bit of a peak, but for some of them, we see absolutely no indication that they're even there. And this could be a problem. If those peaks are above the AET, they still have to be accounted for, and they would be lost out in a base peak mode approach. And it's important to know that the technology exists to do this for LC and GC data. And so I think that long term, really that's what the push is going to be. The expectation will be that you're using extracted ion chromatograms, data deconvolution, to speak to all the compounds above the AET, not just the most abundant that may elute at that particular time. There's also a considerable amount of discussion around chemical identification. So what they're looking for is confident and confirmed. We need a single identity. And that's pretty simple, especially with libraries. A lot of the labs that are doing this, that are kind of on the front end, are really working to build up libraries of the things they see commonly so that they can get confirmed hits on all of that. Tentative, there's been a lot of discussion around this. But the expectations are clarifying, and it's definitely a positive development. Tentative, the saying that's always been around is that it's all plausible structures. So to a chemist, to an analytical chemist in the lab, this is pretty scary because they kind of start doing every possible isomer exercise in their head for a given predicted formula, and you can end up where stearic acid has 10 to the 5th different isomers, right? So if we had to report every plausible isomer in a very skeptical sense, it's impossible. But the FDA kind of clarifies on this. When they say plausible structures, they want a consideration of what the devices are made out of, right? They want to know, use the materials of construction, use surfactants, processing aids, additives, all those things. And they want to see a winnowing down to what's truly plausible, given the chemical history of that particular device. And that's much more reasonable. So the FDA expectation is probably going to be 3 to 5 plausible structures, right? So on the bottom, I've got an example of that, right? It may be, a sulfate or a sulfonate with a hydroxy group, right? But these are all, it's the same formula, but these make sense based on, some sort of chemical argument, right? On the left two, we've got isomers of, a sulfate surfactant, and then on the one on the right, it's sulfonate that's been, that may be present as an impurity in the mixture, right? But that's very different from reporting thousands of compounds for every single identification. And this approach is supported by literature references. So if you look at the broader literature on non-targeted analysis, there's a lot of discussion about this. It's by no means a closed issue, but I think that overall, the FDA is weighing in on taking a direction with this, and that's supporting this Bayesian approach. But this really does reward cutting edge equipment and comprehensive libraries, right? You can narrow down your plausible structures by generating a unique predicted formula, and you can narrow it further down if you've got comprehensive libraries, right? So then you can escalate it from a tentative to I have a library match good fragmentation data or strong interpretation, and come up to confident. More broadly, there's also a drive to qualify methods and then also experimentally demonstrate uncertainty factors. And they are weighing in on this. There's discussion in the literature about a multi-detector approach that Jordy's lab discussed. And regardless of the weighing in on the scientific issue there, the FDA is not currently accepting that argument, and so they really want to see one uncertainty factor per detection method. And this is really important. This is kind of a change in expectation. There are literature values for uncertainty factors that are acceptable, but if you can experimentally demonstrate lower ones, you can use those, and that's really helpful for designing better projects, right? You can cut down on concentration factors, you can cut down on the number of devices you use, with all the impacts for a study sponsor that would have. It's also something worth asking, right? If you're looking at doing work with the laboratory, do they have experimentally demonstrated uncertainty factors in a report that they can deliver to the FDA? Have they qualified all their methods? These are relevant things, and you can ask these at the beginning of the project to make sure that you're on the right track. Lastly, there's discussion of reference standards. The middle responder approach is not recommended anymore. That used to be an acceptable approach, but it is no longer. There's also a drive now that in place of that, they want to see a diverse mix of standards, and that's to enable surrogate semi-quantitation with chemically similar species. What is that going to do for us? That gives us more accurate semi-quantitation And using a multi-standard mix is also going to help us demonstrate method suitability at the time of use. This is something that should all be nailed down in a protocol. And so a lab that's really, investing effort and staying abreast of things is going to be able to answer all these things. Next, I'd like to talk about the chemical list for analytical performance. I'm going to give a bit of an overview and then dive into some of the details on this. So one of the things that's really important about the CLAP list is that it is telegraphing the FDA's beliefs on known unknowns versus the universe of possible compounds. I kind of alluded to this when I was saying plausible compounds in the identification section for the draft guidance, and we can get into that a bit more. So the known list, or the CLAP list, is mostly expected compounds. And we've also seen feedback asking for uncertainty factor justification reports to include relevance to medical devices. So this is giving us some information. They're not interested in really searching for every possible compound that could remotely exist, the universe of compounds, so to speak. They're really interested in what's going to be relevant for medical devices. And the CLAP list also telegraphs to us what a response factor database for uncertainty factor calculations should look like. It tells us some details, right? It gives us some idea on ranges, and it also gives us an idea of what things are considered relevant. And that includes the physicochemical parameters that are used to demonstrate a breadth of coverage, right? So the ones included in the CLAP list are double bond equivalent, molecular weight, boiling point, pKa, log P, and refractive index. Those of you with a toxicology background will recognize these. These are things that are considered relevant in terms of modeling toxicology if we're using, a comparison to a known compound to an unknown compound. These are kind of things that, bioavailability has been pegged to, right? Especially log P, that oxanol water partitioning coefficient. So it's not too much of a surprise, but it's nice to see that this is what they're really interested in. So back to that universe of possible compounds versus known unknowns. The CLAP list tells us what are we really looking for, right? The universe of possible compounds is a very large but not infinite number, and it's neutral on frequency, and it's really the broadest net possible, right? So we're saying if I have a predicted formula, I need to be concerned that every single compound that I can draw that makes sense with that would be stable in air, that I need to be looking for that. In contrast, we kind of have known unknowns, right? For those of you that have been in this industry for a while and looked at a lot of these reports, you know that really there's a few compounds we see all the time, and then as outside of that, as the number of total compounds we're looking at gets larger, we see the additional ones far fewer times, right? We can kind of expect what's going to be there, right? There's a limited number of cleaning agents. There's a limited number of base polymers and additives that people will put into medical devices. And so we can kind of leverage that knowledge to sort of narrow things down and not be so skeptical that we can't really provide any meaningful information. And this is what's meant by the Bayesian approach, right? If I add these things intentionally to the device, I'm probably more likely to see it. And it definitely also encourages us to make sure that we're collecting all the information we can get on what's actually in these devices before we start a project. So again, we're looking at common extractables and we're looking at relevance to medical devices. So it's not stated anywhere clearly this is the official policy, but what we're seeing in the CLAP list and what we saw in the identification discussion in the draft guidance suggests that this balance that I'm showing you here is tending a bit more to the right. We're looking at known unknowns really instead of worrying about the universe of possible compounds. So if we look at the tier 1 list that's already been released by the FDA, this CLAP list, we have a multi-purpose tool. It helps us to understand what types of compounds we should be including to build uncertainty factors for methods. It tells us the physicochemical parameters that are considered relevant. It's that list that I've already discussed. They tell us some concentrations. I find this interesting for people that are in the lab actually doing this work. They know that an LOQ for a method of 5 micrograms per mil is not going to buy you a lot unless you're prepared to do some massive concentrations, which we don't want to do. My understanding is that there's further work planned on this, and that there should be some response factor databases that are lower. in terms of what the LOQ is. So, I'd be expecting, below 1 ppm. And that's where really an analytical lab should be, because they want to minimize concentration, but they still want to make sure their AT is above their LOQ. So there's definitely a drive to get the LOQ as low as possible. And these numbers just on face aren't necessarily where you would want to put it. It's also nice because they do provide chromatography methods. And these include not just the chromatography, but also the detection settings, right? So we've got the mass spectral range and ionization settings that the FDA clearly thinks are reasonable for these types of methods. They also provide a gradient, which again shows what they think is a reasonable approach to this. And this is really good because all the labs doing this work have this information, but there's not really a lot of discussion in the literature on what makes the most sense, right? There's some stuff there, but it's all kind of informal, and there's this kind of feedback thing going on with the FDA and the labs. And it's helpful to get an idea of what the FDA thinks is at least in the neighborhood of reasonable, right? Especially with the mass spectral range. So we can compare to that. We can say, yes, we're on the same page as the FDA, roughly. There may be some small differences, or we can look at it and say, oh, it's drastically different and we need to make some adjustments. So overall, it's good. Again, more information they're kind of providing and can help people evaluate where they are with their processes. So impact. Really, the chemical characterization reports that are being generated for Part 18 studies need to incorporate the chemical list for analytical performance directly or indirectly. And what that means is that for relative response factors to determine the uncertainty factors for methods, really should be incorporating those compounds into your list, and the information as well for coverage. Method validation should also be addressing this, right? A method validation should be should validate a method that's going to be useful for these. Now, I think if someone were to be creating one from scratch, they really need to look at the CLAP list and make sure that they're going to cover that list adequately with their method before they move to validation. And again, it looks like known unknowns appear to be prioritized, and for labs that have been keeping abreast of things, this makes things much clearer as to what the expectation is going to be. So that concludes the discussion of the CLAP list. The last thing I want to talk about is the accreditation scheme for conformity assessment workshop. So this was held last November by the FDA at the headquarters outside of Washington, D.C. And it was well attended, and it opened with a discussion of the round robin study that the FDA prepared and had out. Some of the conclusions from that, of course, were, wow, more work needs to be done on this. We could get into those details, but it's not really within the scope of this talk. So I'll just say that there was discussion of that, and I expect that there's going to be more movement from the FDA on that, perhaps maybe another round robin or series of round robins. It's my interpretation. There was also kind of a clear statement that the ASCA accreditation is really, it's going to be executed in the following format: is that accrediting bodies will be accredited and approved by the FDA. And then those accreditation bodies will then do that for labs. And the current plan is at least to only roll that out for labs in the United States for now. So that may influence the decision-making process for choosing labs to do work when this is done. So none of this has been done yet in terms of accreditation. It's just in the discussion phase of what that's going to look like. So let's talk a little bit more about those things. The topics they were really interested in were method validation, response factor databases, and uncertainty factor determination, gravimetric NVR. What would a summary test report look like, and personnel competency evaluation? And this was really good because we had a lot of the major players in performing this work in the room with the FDA, and they were discussing it in a neutral setting, in the sense that we weren't talking about particular projects. No one was really worried about the particulars of a specific device that was under debate. It was just a general discussion of approaches. And I think that was really good, because we need that kind of more general discussion, and then the clarification of FDA expectations that are informed by industry experience. So there was discussion around what method validation and demonstration of that would look like, as well as response factor database, uncertainty factor database determination. It's a discussion of how to demonstrate the breadth of coverage for the compounds used to determine the uncertainty factors. I don't think that's a closed issue yet, but it's a good discussion to be having. And I think that an uncertainty factor determination report should probably have some sort of demonstration of that breadth. At its most basic, it can be that it includes the CLAP compounds as a minimum, but there can be additional things that could be done. There was discussion around gravimetric NVR and the challenges there because gravimetric approaches just simply aren't as sensitive as modern chromatographic ones. It's a little bit of it can be a little misleading. There are some very precise and accurate microbalances available on the market, but buying those and trying to do non-volatile residue analysis on them, it starts to get into statistical noise from other parts of the process. And there was a clear back and forth about that at the meeting, and I think that was good. A summary test report, the idea itself is excellent. It remains to be seen what exactly that'll look like, but the idea is kind of just creating a coherent and consistent test report format that's going to streamline the review process. And lastly, there's discussion of personnel competency evaluation. In related industries where we're doing targeted work, it's a little bit easier to evaluate that, right? We can send in something of a known concentration, have it evaluated. And either you get the right number or you don't. You can set acceptance criteria pretty easily because this is all a quantitative approach. For non-targeted analysis, that's not necessarily as straightforward, and there's discussion around that. I think everyone knows that this should happen. The question's really just in the details. So let's wrap up with some conclusions. The first conclusion that I would like everyone to take from this is that the FDA is clarifying expectations for chemical characterization, but it's still an evolving situation. For those of us who've been in this for a while, that's kind of the way it's been, is that it's always been evolving, and that, you know, a project done 2 to 3 years ago may not be done the same way as one that is started today. But it's good because they are clarifying expectations of what they want to see for that. I will say, based on the ASCA meeting as well as you know, other sources, that response to these recommendations across the industry may be variable. And I'll get into that a little bit more in a second. But on the upside, the forward-looking organizations are seeing their practices reflected in the guidance. You know, if an organization is reading what's out there and they're paying attention to the feedback that's coming from the FDA on the projects that they're working on, if they're participating in standards committees, if they're reviewing the literature, these sorts of things, and they're really working hard to do the best work they can and innovate, what they're actually seeing is they're seeing that, you know, well, the practices we're doing are actually now being reflected in the guidance. It's becoming an expectation. So the response to that can be variable. And the reason is that ISO 10993 Part 18 is becoming more specialized. When this work first started, it was possible you could go to a lab, and this may be a portion of the work they do, and they do a lot of other work because there was more overlap. But really, now that these expectations are being clarified and more specific approaches are being defined, really what we're seeing is a divergence point between other types of lab work and this non-targeted analysis. So as it's more specialized, you know, laboratories have to reevaluate their participation in it. If they've been doing it and they have not been updating instrumentation and processes, it may be a costly endeavor to modernize, right? And this could definitely be the case for internal labs that maybe do lots of work, and this is a small portion of what they're doing, and they're trying to use their general tool set to do it. To catch up, you know, to be able to meet the data deconvolution expectations and other parts of that approach, it may cost a lot in terms of, you know, all resources. And so it may be in some cases that it actually makes more sense to begin outsourcing that work, or you know, or go through the costly endeavor to modernize. So overall, this is good stuff to be going on, and ultimately it's going to have a positive impact for patients, and that's, you know, that's why we're all here. But it's definitely a lot of details and a lot of things to keep track of in a highly functional environment. So I want to thank you all for your time today and kind of sitting down and listening to a discussion of a lot of these recent developments that have been going on. And I just want to say that you can always contact us at the URL below if you'd like more information. Thank you.